Well, a warm welcome to this talk. It's Sunday, the 30th of April. Now, today's study is really quite impressively interesting. It's about cannabis. Now, most of you will probably know in cannabis there's two main ingredients. There's the THC, the tetrahydrocannabinol. That's the bit that gets you high if you use this substance. And there's the other one, the CBD, the cannabidiol, which is very much psychotropic. It doesn't affect the brain at all. Now, I'm going to give evidence today that the CBD, the cannabidiol, seems to have, and there's pretty good evidence from this study and other studies, antiviral properties. Now, we'll be looking at evidence that it reduces the infection from SARS coronavirus 2 and helps with the illness. And we'll be looking at evidence that it helps with another RNA virus, hepatitis C. Now, let me tell you what evidence I'm going to be considering, and you can decide if you want to watch this video. This is oral, by the way. It's not injections. And in all cases, it's been completely non-toxic at the doses that have been described. Now, the first line of evidence that this works against SARS coronavirus 2, and probably other viruses, but the research is on SARS coronavirus 2, is in human lung cell cultures, where it prevented infection. When the cells were infected, if they'd been exposed to the cannabidiol 2 hours before, it essentially prevented all of the infection. But it didn't work with THC. So this means that if people are smoking cannabis or using cannabis products that are mixed with CBD and THC, the ones that will get you high, it won't work. This is strictly the non-psychotropic version. Tetrahydrocannabinol inhibited the effect of the cannabidiol in terms of antiviral properties. So that's the first one, human cell cultures. The second evidence is from mice, so it actually works in animals. Infected mice, they were given CBD, cannabidiol twice a day for 4 days, and they had 40 times less viral particles in their lungs. Really quite impressive. And the third line of evidence is human. Now, in the United States, they managed to recruit 1,221 people that are on cannabidiol to prevent epilepsy. And they found that they were 50% less likely to get infected with SARS coronavirus 2. And they had no follow-up on whether they got sicker or not, but it would be likely that these people didn't get sick as their counterparts may have done, especially with the early versions of SARS coronavirus 2. So 3 impressive lines of evidence we're going to look at. Probably works against hepatitis C, some evidence for that. Terrible disease, hepatitis C. Could well work against colds, flu, Ebola, mumps and measles. The reason I'm saying that is purely because there are other RNA viruses. We don't know, but it could potentially work against that. So I would certainly call for immediate research to be done on this. But of course, hemp plants, which is where this essentially comes from, can be grown up by the hectare. And hemp oil, the cannabidiol, can be produced for essentially nothing. And it can be produced by anyone. It's not a specific preparation. So again, because any producer who meets basic food manufacturing requirements could set up and produce this, it's unlikely that Big Pharma are going to do the clinical trials. So this could be another one of those potentially brilliant treatments that's going to be completely lost to humanity because there's no money in it. And I'm going to stop there, otherwise I'll get cross. Let's go back and look at the evidence now. That's what this video is about. So this is the original paper here. Cannabidiol inhibits SARS coronavirus 2 replication and promotes host innate immune response. Now, this was a preprint. So I did come across this, I think, last year, but I decided not to take it up. But now it's fully peer-reviewed, so we now have the evidence in hand. And well, it's taken me a while to read through this paper. It really is quite an impressive piece of work. Very biochemical. I had to look up a few other words, I must confess. But so this is my understanding of the paper. But I think I've got the main points fairly accurately. Cannabidiol, potential regulator of the host stress. So this is the way that the body responds, or the cells respond to viral infection. And it also looks like it helps antiviral inflammatory responses. Now, there was 2 things here. First of all, it helped the beneficial inflammatory response, which is what you need as part of the immune response. But also, it inhibited the release of the cytokines, which causes the cytokine storm. So it looks like we're getting the benefits of the immunity, but inhibiting the cytokine storm. So it really is a win-win. preparation, according to the evidence cited in this study. Cannabidiol class of natural products, so it's a natural product. Natural products, of course, are great. There's all sorts of natural products. Let's think of some, for example, opium is a natural product from opium poppies. Antibiotics are a natural product from molds. A lot of the best therapeutics come from natural products. Others we could mention. It's an oral solution, so that's good. Now, hemp plants contain less than 0.3% THC, tetrahydrocannabinol. So, but we'll show that this preparation was actually fairly pure CBD. Oral solution is already FDA approved. So given that this is FDA approved and it exists, there's no reason why, in theory, doctors shouldn't be allowed to prescribe it. It would be off license for this product because it's only licensed for epilepsy at the moment, or various types of fitting. And as I say, the large-scale clinical trials probably won't be done, but they should be done. High purity. CBD inhibits SARS-CoV-2 replication in human lung epithelial cells. So this is human cell lines that they're taking. Cells that were taken from humans have been bred up in cultures, and they're able to do experiments on these. In my view, this is quite ethical research. It's actually human cells taken from people who had lung cancer, is where they got it from. So they pre-treated human cells with CBD, and these are with the ACE receptor. For 2 hours, so the pre-treated with CBD for 2 hours, CBD potentially inhibited viral replication under non-toxic conditions. So the doses here that they used, if you scaled this dose up to the whole person, they were non-toxic doses. And CBD is a safe preparation anyway. Well, as safe as you can never say anything. It's completely safe, but it's basically safe. You can certainly buy it in supermarkets in the UK, for example. Worked in other human cell lines, so they tried it in other human cell lines. They also tried it in monkey kidney cells. It also worked. They tested it on the Wuhan strain and 3 other variants of concern, and it also worked. The ability to infect cells was comparably inhibited by cannabidiol. So in all of these, the Wuhan strain, the 3 variants of concern, all of them couldn't effectively or as effectively infect the cells treated with cannabidiol. So it looks like it's working for all of the SARS-CoV-2 viruses. And as we say, very likely to work for other viruses. Now, they tried quite a few. They tried a few preparations of cannabidiol to make sure it wasn't just the particular preparation. So here they had different suppliers. Here they have the positivity of the spike protein, and we see here that as the doses go up, the numbers go down until up in these doses here we essentially have no viral expression. And as we say, these are non-toxic doses that you could have used. And they actually tested these with various methods to make sure that it was fairly pure cannabidiol, and they found out it was 97 to 100% pure cannabidiol. Because when you extract products from natural plants like the hemp plant, you're going to get other alkaloids in there as well, other cannabis products as well. But they were sure it was the CBD and one of its, as we'll see, one of its metabolites that was having the benefit. So was it the other cannabinoids? Well, no, they checked. So basically it wasn't the other cannabinoids. 97 to 100% pure using quantitative nuclear magnetic resonance scanning. Don't pretend to know much about that at all. But it is an industry standard way of measuring compounds in plants and things like that. So it is an accurate way to do it. That is clear. No toxicity was observed for any of the CBD preparations at the doses used to inhibit viral replication. No toxicity. This was safe at the doses used. It could be scaled up to the whole human body. And the CBD metabolite, so it was CBD and its metabolite, 7-hydroxycannabidiol. Now what happens here is this is quite common. For example, if you take codeine, for example, the codeine itself isn't a very good painkiller, but it goes to the liver, and the liver converts it into morphine. So the reason that the reason that codeine is an effective analgesic is the metabolite is morphine. And in the same way here, the CBD worked, and one of its metabolites, the 7-hydroxycannabidiol, also was antiviral. So two ways to win there. The CBD probably working originally when you take it, then some of that's broken down into this 7-hydroxycannabidiol, and that goes on working for a period of time. So, and interestingly, these are exactly the same molecules that are known to be effective in suppressing some forms of seizures that people take cannabis for already, or this particular CBD preparation for. So both of these, the CBD and its metabolites, exhibited antiviral activity. The other cannabis compounds did not work, and combining CBD with tetrahydrocannabinol significantly suppressed CBD activity. So taking the sort of cannabis that gets people high would not work. That's important to stress. Taking the cannabis that gets people high would not work, because it basically there's a competition between the THC and the CBD, and that stops the CBD working. The CBD and its metabolite are the active and equipotent ingredient for the treatment of epilepsy. So that's why it works. So the same things that work in epilepsy are the same things that are in this newly discovered antiviral, this very impressive antiviral property. So it's working by early steps after viral entry into cells. It's reducing that. CBD targets host cell processes. CBD exhibits viral RNA expression and reverse virus-induced changes in the host genome. So, in other words, the virus, actually, what the virus does is it changes quite a few things in the cytoplasm and the nucleus of the cell to make it more hospitable for itself. The CBD undoes those changes. Quite amazing, really. I mean, I don't pretend to understand how it does this, but they did identify quite a few mechanisms that can work for that. CBD effectively eradicated viral RNA expression in the host cells. I mean, wow. They weren't getting the viral expression. The virus just couldn't reproduce inside those cells. Prevents changes in the cell produced by the virus to force the cell to produce new viral particles. So the virus has got all these mechanisms to take over. The CBD blocks those. CBD produces in strong activation of genes associated with host stress response. Now, the stress response is the way that the cell responds to the infection, and the CBD initiates the activation of genes which help that response. So, if you like, the CBD is partly working by genetically upgrading the cell by stimulating the genes to make more antiviral activity in the cell. Quite amazing, but this is what the biochemistry shows. Direct quote from the paper: together these results suggest that CBD acts to prevent viral protein translation and associated cellular changes. Amazing. Induction of viral genes for spike envelope and nucleocapsid protein is reduced by 99%. Just incredible. Just incredible. So if this is demonstrated to be effective as it looks, if everyone in the Wuhan area or around the people that were first infected, won't go into the story of that now, but if they'd been on CBD, that could have been a firewall around about this virus, and a pandemic might have never occurred. Non-toxic, simple, cheap, readily available by the ton. Nothing complicated, no huge profits for anyone. And of course, if it had been eradicated at source, we would have never have needed the vaccinations either, because there would have been no pandemic. CBD induction of increased transcription of 3 defense genes, as we've said. And these affect the endoplasmic reticulum of the cell, which I won't go into, but it's part of the cell that's associated with the ribosomes, which makes the new viruses. It stops those from working. Also, CBD induces interferons. We've talked about interferons before. Interferons are antiviral cytokines, really, antiviral signal molecules that when a cell is infected by a virus, it can release interferon. And that goes to the cells around about and tells them to prepare for the infection. So it kind of puts a firewall around about the infection. So stimulating interferons also very important part of protection against viral infection. Promotes degradation of viral RNA, so it messes up the viral RNA. SARS coronavirus 2 infection suppressing the interferon signaling pathways. So the SARS coronavirus 2 would interfere with the interferon activity. This undoes the damage done by the SARS coronavirus 2 and allows the interferon to carry on, and indeed stimulates the interferon. CBD effectively lowers the viral titers, so the viruses aren't there to add their suppression to the interferon as well. CBD effectively reverses viral-induced cytokines that can lead to the deadly cytokine storm. So if this was given to... So this work is about early infection, but it sounds like it's also could have saved many, many lives by preventing the cytokine storm, which is, of course, what killed people, causes the acute respiratory distress syndrome. Really quite amazing. Look at this paper. It really is quite an impressive array of effects that you wouldn't have expected. CBD treatment significantly inhibits SARS coronavirus 2 replication in mice. So the mice were deliberately infected with a virus, a viral challenge. They were given CBD twice a day for 4 days. And CBD treatment significantly inhibited viral replication in the lungs and in the noses at 5 days. CBD lowered dose. So the lower dose of CBD, if they gave a lower dose of CBD, it reduced the viral dose in the lungs by 4.8-fold. Higher dose, 40 times less virus in the lungs. And, you know, for most people, that would be the difference between getting sick and not getting sick. This is really quite staggering. And even the higher dose was a non-toxic dose. It was not making the mice sick. The mice did not stop eating. They did not lose weight. The mice did not get sick, but they had 40 times less virus in their lungs. Looking like a very safe way of doing this. During this period, the mice showed no signs of clinical disease and didn't get sick from the drug. The authors say these results establish the preclinical efficacy of CBD as an antiviral drug for SARS coronavirus 2 in the early stages of infection, but potentially in the later stages because of the reduced cytokines. Now, what about humans? CBD is negatively associated with indications of SARS coronavirus 2 infection in patients' medical records. Now here, what they did was they looked up these more than 1,200 patients who were on CBD already and found that they got less infection. They didn't follow them up when they got sick, unfortunately, but it would be interesting because it would be likely that they didn't get sick as much because they weren't getting infected as much and the viral load would be lower. Of course, all these things should be done with full-scale clinical trials, but as we said, that's probably not going to happen, unfortunately. Given that high-purity CBD preparations are taken by large numbers of individuals, we examine whether CBD prescriptions are associated with interactions of SARS coronavirus 2 infection. Now, apparently, people are taking oral solutions of CBD at 100 micrograms a mil. Often used in the treatment of seizures. They analysed 1,200 patients taking it already. They then made up a control group matched for age, sex, all the variables. They found that 75 patients with CBD, 6.2% of the patients, 75 patients with CBD got infected, 8.9 for non-CBD patients. Now, this doesn't sound like a big difference. 6.2 got infected with CBD, 8.9 got infected without CBD. But of course, this is absolute risk for the whole population. So this is a huge difference. And it worked out. So the odds ratio, in other words, it was a 45% protection for people that were probably taking it. But for people that were sure taking it, it was a 52%, the odds ratio of 4.48, 52% protection. And again, these results are very significant. Only 6 chances in 1,000 that arose by chance. So these people were not getting infected. This was having about 50 people taking the adequate dose were having a 50% protection against infection. It really is quite significant. Pity they didn't have follow-up data on if they got sick or not. But as we say, that work wasn't outside the scope of this study. Potential for the cannabidiol for treatment of viral hepatitis. That's the study there. It's looking good. Here it was CBD inhibited hepatitis C viral replication by 86%. Didn't work for hepatitis B. Now, this is interesting. Hepatitis C is an RNA virus. Hepatitis B is a DNA virus. So it looks like the data we have that this is working for RNA viruses, probably not for DNA viruses. But if we could get rid of the RNA viruses effectively, and have had an effective treatment for RNA viruses, that would help, to put it mildly. Let's just look at some of these infections. Common colds, whether it's rhinoviruses or coronaviruses, are RNA viruses. Influenza is an RNA virus. SARS-1 and Middle East Respiratory Syndrome. Dengue, terrible disease. Hepatitis C and E, all RNA viruses. Ebola, rabies, polio, mumps, measles, all RNA viruses. If this turns out to be an effective treatment for RNA viruses, this would be a major, massive step forward in medical practice. So I think what we need to do is take these clinical trials out of the hands of Big Pharma, because, as we say, I don't think they're going to do it because you can't make money out of CBD. But this is a drug which could be precious for humanity. Therefore, governments and other agencies need to take on this research, and we need to know definitively if this works. And if it does, it could be a wonder treatment for many conditions. Let's hope this is done. The work we have so far is impressive. I'm really quite impressed with this article, as you can probably tell. We'll leave it there because that's as far as the data goes. Thank you for watching.